Welcome. In this lecture, I shall explain epigenetics and its connection to natural selection because this topic has a huge impact on upcoming researches in the field of genetics. In this short video, I shall make you to learn about epigenetics and natural selection, how epigenetics works, some very interesting case studies, and interaction between epigenetics and natural selection, some very interesting specific examples to understand the concept, and finally some future implications. Let's start. What is natural selection? Natural selection is the fundamental mechanism by which species adapt and evolve over generations. The principle of natural selection is that individuals with certain traits that provide them with a reproductive advantage are more likely to survive and pass on those traits to their offspring. This process leads to the accumulation of advantageous traits in a population while unfavorable traits become less common. We know that genetic information is stored in the form of DNA within our cells, and DNA consists of sequences called genes which contain instructions for the production of proteins. These proteins play crucial roles in the functioning and development of organisms. So, the process of selection of fittest in nature to survive and strive is called natural selection. So, what does epigenetics deals with? Epigenetics explores heritable changes in gene activity that occur without altering the underlying DNA sequence. I repeat, without altering the underlying DNA sequence. It involves modifications to the structure of DNA or the proteins associated with it, influencing how genes are turned on or off. Unlike mutations that alter the DNA sequence itself, which are irreversible, epigenetic changes are reversible and are influenced by environmental factors mostly. Now, how epigenetics works actually? There are several epigenetic mechanisms, but let's focus on the two most well studied: DNA methylation and histone modification. Number 1. DNA methylation acts as an on-off switch. DNA methylation involves addition of a chemical group, methyl group, to the DNA molecule, typically at specific regions called CPG islands. Methylation acts as a switch that can turn genes on or off. When a gene is methylated, it is usually silenced and its expression is reduced or completely blocked. Number 2. Histone modification. Histones are proteins around which DNA is wrapped, forming a structure called chromatin. Histone modification refers to chemical changes in the histone proteins affecting how tightly the DNA is wound around them. These modifications can either promote or inhibit gene expression also. Case study 1. Dietary factors have been shown to induce epigenetic modifications. For example, studies have demonstrated that a pregnant mother's diet can influence the epigenetic marks on her child's genes, potentially affecting their health outcomes later in life. These findings highlight the interplay between environmental factors, epigenetics, and natural selection. Case study 2. Studies have shown that individuals who experienced significant stress during early childhood, such as neglect or abuse, can exhibit changes in DNA methylation patterns. These modifications can affect the expression of genes involved in stress response and can have long-term impact on an individual's mental health and resilience to stress later in life, that it may alter the personality. Now, you understood the link between epigenetics and natural selection, how epigenetics interact with natural selection to shape the evolution of species. Let's learn. In this part, I shall explain 4 aspects of interaction between epigenetics and natural selection: phenotypic plasticity, epigenetic inheritance, rapid adaptation, and evolutionary trade-offs. Number 1. Phenotypic plasticity. Epigenetics allows organisms to exhibit phenotypic plasticity, which means they can produce different phenotypes in response to environmental cues. This flexibility enables individuals to adapt to changing conditions without altering their genetic code. Natural selection acts on this phenotypic plasticity, favoring individuals whose epigenetic modifications enable them to thrive in their specific environments. In honeybee colonies, for example, the diet of developing larvae determines whether they will become queens or worker bees. Very important. The queen larvae are fed a special diet called royal jelly, while the worker larvae are fed a different diet. This difference in diet leads to distinct phenotypic outcomes. Epigenetic mechanisms such as DNA methylation have been found to regulate the expression of genes associated with caste differentiation in honeybees. Number 2. Epigenetic inheritance. Epigenetic changes can be passed down from one generation to the next. This transmission of epigenetic information is called epigenetic inheritance. It provides a means for acquired traits influenced by the environment to be passed on to offspring. Consequently, natural selection can act upon these inherited epigenetic changes, leading to adaptive responses over time. Number 3. Rapid adaptation. Epigenetic modifications can occur relatively quickly, even within a single generation. This rapidly allows organisms to adapt to new or changing environmental conditions more rapidly than would be possible through genetic mutations. This swift adaptation increases the chances of survival and reproductive success of individuals and contributes to the overall diversity and resilience of species. Researchers studying a population of soy sheep on the Scottish island of St. Kilda found that during harsh winters with limited food availability, certain genes involved in metabolism and energy utilization were epigenetically modified, allowing the sheep to conserve energy and survive under challenging conditions. This rapid adaptation through epigenetic mechanisms provided the population with a survival advantage. Number 4. While epigenetic modifications can confer advantages, they may also come with trade-offs. For instance, a particular epigenetic change that enhances an organism's resistance to a specific pathogen may simultaneously impair its ability to cope with other stress. These trade-offs can influence the direction and extent of natural selection, leading to complex interactions between genetics and epigenetic factors. Let's consider an example. Parental investment refers to the resources and care that parents provide to their offspring. In some species, such as birds, the level of parental investment can have a profound impact on offspring phenotype and survival. Epigenetic mechanisms can regulate the allocation of resources and influence trade-offs between investing in current offspring versus future reproduction. For instance, a study on great-tit birds found that the maternal diet during egg formation led to epigenetic modifications that affected offspring size, highlighting the trade-off between current reproductive success and future offspring quality. Finally, let's talk about some future implications or impacts of this area of genetics. Evolutionary biology. The integration of epigenetics into evolutionary biology has opened up new avenues for research. Scientists are now exploring how epigenetic changes can thrive or drive evolutionary processes such as speciation, adaptation, and response to environmental challenges. Number 2. Personalized medicine. The field of epigenetics holds promise for personalized medicine By understanding an individual's epigenetic profile, it may be possible to predict disease susceptibility, tailor treatment strategies, and develop targeted therapies that can modulate specific epigenetic marks. Hope you learned new things. If yes, do like and share it with your close ones. Thanks.